Hello guys, welcome to Molecular Biology series. I'm Vineet and today I'll be discussing about the packaging the eukaryotic genome or the uh, condensation of this long thread DNA into compact sized chromosome structure. Okay, so how this condensation happens. Okay, so if you look at the overview of this genome organization inside the eukaryotes, okay, it basically involves a lot of different types of proteins. Okay, so one major protein groups is the histone proteins. Okay, there are two different proteins, core histone proteins and the linker histone proteins. Okay, so these histone proteins, they contains a lot of basic amino acids or the positively charged amino acids, lysine and arginine. Okay, so due to this presence of the lot of basic amino acids, these histone proteins, they contain positive charge on them. Okay, so these are positively charged proteins. Okay, so DNA being negatively charged molecule, it readily wraps around this histone proteins due to this electrostatic attraction forces, and they form a specific structure known as the nucleosome. Okay, so this nucleosome is actually made up of a long thread DNA, which is around 167 base pairs or 200 base pairs DNA sequence, along with 8 histone proteins. Okay, so most of these histone proteins, they will be presenting as dimers. Okay, they will be present in this nucleosome core as dimers. Okay, so these nucleosomes, okay, they further gets linked with another different types of another nucleosomes. Okay, so they form a bead sounding structure. Okay, so the DNA containing the nucleosome core. Okay, so when you observe this form of DNA under the microscope, okay, it appears like a bead sounding structure. Okay, so these nucleosomes, they further condense to form the next level structure, that is the 30 nanometer fiber. Okay, so this fiber is often referred to as the solenoid model of the chromatin, and this is actually the uh, second level condensation, second level of this condensation, second level structure of this condensation process. Okay, and most of the genes are inactive in this form. Okay, and this 30 nanometer fiber further condenses with the help of this scaffold proteins. Okay, which you also called as the non-histone proteins. Okay, so they are the SMC group proteins and topoisomerase 2 alpha. So these kind of scaffold proteins, they further condense this 30 nanometer fiber and make this into a specific chromatid molecule. Okay, and the further condensation in the metaphase at the end of this prophase. Okay, so you will be seeing the whole compact structure of this chromosome. Okay, along with the centromeres and the kinetochore proteins. Okay, so this is just an overview of this genome organization or the condensation of the DNA molecule into compact sized chromosome. Okay, and if you look at the first level of chromosomal DNA packaging, okay, so that is referred to as the nucleosome. So this nucleosome actually contains the proteins along with the DNA. Okay, so the continuous DNA fiber of each chromosome is packaged into many hundreds of thousands of nucleosomes. Okay, so that means per cell nucleus, okay, you will be seeing around millions of, okay, so around 3.3 millions of nucleosome structures inside the nucleus. Okay, so around 3.3 millions of nucleosomes are present. Okay, so this long thread of DNA is wrapped around this histone core proteins. Okay, that is what makes the nucleosomes. Okay, so individual nucleosome can be isolated by following the cleavage of DNA between neighboring particles by nucleus. Okay, these nucleus are also called as the major nucleus, which we use for the nucleosomal extractions. Okay, so separation of this individual nucleosomes. Okay, that is called as a micrococal nucleus, or you can also call that as MNAs. Okay, so they digest the chromatin material randomly. Okay, and yields a mixture of short chain nucleosomes. Okay, so which you also refer to as the compacted nucleosome structures. Okay, and further digestion with the nucleosomes, yields individual nucleosomes. Okay, so continued nucleus cleavage. Okay, so the continued nucleus cleavage yields a stable particle around 146 base pairs of the DNA along with the proteins. Okay, so this 146 base pairs of DNA, what you actually call as a typical nucleosome or the single nucleosome core. Okay, so the nucleosome core particle. Okay, and then this nucleosome core particle actually contains an octamer of proteins. Okay, that means eight proteins. Okay, so the two closely linked heterodimers, which you also call as H3 H4 heterodimers. Okay, and then we have H2A H2B heterodimers. Okay, these two heterodimers, they form an octamer. Okay, So what we have here is, two H2A H2B heterodimers. That means 2 into 2 proteins. Okay, that forms 4 proteins. Okay, and here also we have what, two closely linked H3 H4 heterodimers. Okay, 2 into 2, 4 proteins. Okay, so these 4 plus 4 proteins, they form a octamer core of a set of 8 proteins. Okay, so these 8 proteins along with the DNA is what you call as a nucleosome core particles. Okay, and we also have another histone molecule which we often call as a type 1 histone molecule, or the linker histone. Okay, which connects the uh, entry and exit site of the DNAs. Okay, so this is what about the nucleosome core structure, which is made by histones as well as the DNA molecules. Okay, and if you talk about the number, human nuclei contain roughly 3.3 into 10 power 7. That means 3.3 million nucleosomes distributed along the DNA molecule. Okay, so that a typical nucleus contains around 3.3 nucleosomes. Okay, and the structure. Okay, so this is what the the beads on string structure when you observe under the microscope. Okay, the during the interphase stage. Okay, so in the interphase of a cell cycle, okay, if you observe the interface under the microscope, you will be seeing this kind of structures, where the DNA is rapidly replicating, or performing some kind of gene transcription things. Okay, and if you digest this chromatin molecule with the MNAs, the nucleases. Okay, so on initial digestion, it will yield a nucleosome containing 200 base pairs of DNA sequences. Okay, and continued digestion by these nucleases. Okay, they will yield 146 base pairs. That is around 1.76 tons of DNA around this nucleosome core. Okay, so we have a nucleosome core over here, a set of 8 proteins. Okay, so the DNA actually wraps around 1.76 tons. Okay, so this typical nucleosome core contains around 146 base pairs. Okay, so continued digestion will give you the 146. Okay, the initial digestion you will be getting 200 base pair DNA sequence around the histone core proteins. Okay, so this is what about the nucleus digestion and the structure of the nucleosomes. Okay, if you talk about the actual structure. Okay, so we also have this histone linker histone over here that connects the entry and exit site of the DNA. So okay, this is the DNA entry site into the nucleosome core, and this is the exit of this DNA sequence. Okay, so this DNA thread, when it enters, okay, so the histone proteins allow the DNA molecule to bind over it or to wrap over it. Okay, and the same DNA, after making 1.76 turns, okay, it will leave the histone core particle. Okay, so this entry and this exit. Okay, so it is bounded by a typical histone called as a histone H1. Okay, linker histone. Okay, so this linker histone is not part of a histone core. Okay, so this is a separate protein. Okay, so if you remove this linker histone, then the genes which are present in this DNA sequence will be active. Okay, if you add this linker histone, then it will convert into heterochromatin state. Okay, where the genes will be in an inactive form. Okay, and if we even look at the diameter of this entire nucleosome core model, that is around 12.12 nanometers. Okay, that is around 10 to 12 nanometers. Okay, the number is not fixed because a lot of different textbooks uh, gives different numbers of diameters. Okay, the some textbooks, I've seen 11 nanometer diameter, but in some, the size of this nucleosome is around 12 nanometer diameter. Okay, some textbooks also have given this 10 nanometer diameter. Okay, and this simple, the nucleosome model, okay, the whole nucleosome model along with the histone molecule contains 166 base pairs of the DNA. So that is approximately full 1.98 turns of the DNA molecule. Okay, so that you can consider as a 2 full turns. Okay, and without the histone linkon, without the linker histone, you will be seeing only 146 base pair nucleosome model. Okay, that means only 1.76 turns of DNA is present around the nucleosomes. Okay, so this one without histone linker histone, okay, and 166 base pairs or 2 full turns, that describes entire nucleosome models along with the linker histone molecules. Okay, and if you look at the molecular weights of this linker histone and the nucleosome model, okay, and the DNA sequences, that is actually same. Okay, so if we look at this, the molecular weights of this histone proteins and the DNA sequences. 200 base pairs of DNA approximately weighs around 130 kilodaltons, okay, and the total proteins, okay, the histone octamer, the core nucleosome contains molecular weight around 108 kilodaltons, okay, and if we even include this linker histone, that linker histone weighs about 24 kilodaltons, okay, whole together, it will be 132 kilodaltons, okay. So this 132 kilodalton weight of this proteins and 130 kilodalton weight of this DNA is almost equal, okay, and the equal amounts of this or equal weights of DNA and histone makes the nucleosome model, okay. So the nucleosome model contains actually equal amounts or equal weights of DNA as well as the proteins, okay. So this is what the basic things about the nucleosome model, okay. And I've said you this nucleosome model actually a representation of the solenoidal form of this DNA calling, okay. So the DNA wraps around the central axis. Here the central axis is what the histone octamer core. So this is the central axis which you also called as the central octamer, okay. So this on this central octamer the DNA will wrap around it, okay. This is what gives you the toroidal model or the super solenoidal model of the DNA, okay. So this kind of structure, okay. So this is called as a solenoidal wrapping of the DNA in nucleosomes, okay. So whenever one nucleosome connects with another nucleosomes, okay, so the amino terminals of this histone proteins, okay. So histone proteins are made up of proteins contains the amino terminal as well as the carboxy terminal, okay. We all know this, okay. So this amino terminals of these histone proteins, they extrude out from the nucleosome cores, okay. So this is what the point is, okay. If you look at this point, the amino terminal tails of one nucleosome extrude from the particle and interact with adjacent nucleosomes, okay. So these kinds of attachments or this kind of interaction helps defining higher order packing of the DNA, okay. So further condensation of the DNA will be very easier if the amino terminals of this nucleosome or histone proteins if they interact with the other histone proteins, okay. And these amino terminals they also involved in the regulation of gene activities, okay. Lot of methylations and the acetylation things happens on this amino terminals, okay. So methylations and acetylations will regulate the gene activities and they are also involved in the epigenetic regulations, okay. And if you even talk about the molecular weights of histones and the contents, okay, amino acid contents of these histones, okay, I said you already the molecular weights is around same as DNA molecular weight, okay. But this amino acid contents, okay, lysine, arginine, okay, they vary between histone to histone. Histone 3, histone 1 actually contains higher amounts of lysine and arginine, okay, that is around 40 42% of basic amino acids, okay. That is 42% of basic amino acids will be present in this linker histone or H1 histone, okay. And around 32% or around 33% of basic amino acids will be present in this H3 histones, okay. And most of the modifications will be taking place on this histone 3 only, okay, at 4th position and the 9th positions, okay. So that makes the higher gene regulation activities inside the nucleus, okay. Okay, so this histone 3 is majorly involved in the gene regulation activities, okay, and then the histone 4 and H2A and all these things, okay. Most of the gene regulation activities won't be seeing in the case of the histone 1, but even though it contains a lot of basic amino acids, okay. And yeah, one more point what you have to remember is the cluster of 280 base pairs, okay. That means for every uh, 10 base pair repeat, okay, so for every 10 base pairs, okay, so presence of this 80 base pairs, okay, cluster of 280 like this, okay. So they promote the bending of the nucleic acid chain over the histone proteins, okay. In order to make your DNA or in order to make the nucleic acid chain to bend over this histone proteins, okay, to wrap around this histone proteins like this, okay. So this bending movement or this wrapping movement is facilitated by the clusters, okay. These clusters are mainly rich in the adenine and thymine, okay. So since they contain only double bond between them, okay, so they can easily bendable and we can wrap the DNA around the histone proteins, okay. And the presence of GC pairs, okay, so they will do the opposite thing. Okay, they will make the DNA strand to be in the linear form. Okay, so this is what facilitates the nucleosome model. Okay, so the one thing that facilitates the formation of nucleosome model is in a presence of 80 base pair clusters. Okay, 2 or 3 80 base pair clusters for every 10 base pair intervals. Okay, you have to remember this point. This is very, very important. Okay? And this diagram actually tells about the bending of the DNA in nucleosome. Okay, so this bending is actually facilitated by what? Double A, double T, or adenine and thymine dinucleotide clusters. Okay, repeats. Okay, for every 10 base pairs, your nucleic acid must contain this adenine thymine clusters. Okay, so that this DNA will interact with the nucleosome core proteins. Okay, so at this areas. Okay, this 80s or the thymines or the adenine sequences, they interact with the nucleosome model because they only have the double bonds between them, and they can easily bendable. Okay, malleable. And the GC, they form the major group, so that means they protrude out from the nucleosome core. Okay, so they actually quite opposite to this 80 reach sequences. Okay, whatever the bending thing is happening over here. Okay, so that is enhanced by these 80 sequences, while that is opposed by the GC sequences. Okay, so the opposing the bending thing is done by this GC, and enhancing the bending thing enhancing the bending moment of this DNA is enhanced by the adenine and thymine and thymine adenine nucleotides. Okay, so for every 10 base pair intervals, the nucleic acid should contain this dinucleotide repeats. Okay, adenine thymine or adenine or thymine. Okay, while guanine and cytosine or guanine or cytosine sequences, they are involved in formation of the minor group. Okay, or minor group or major group. Okay, so this is what the typical DNA sequence will look like around the nucleosome core. Okay, so if we consider this is a 1.76 base pair turn. Okay, so and this is the entry site and this is the exit site. Okay, if you could observe, for every 10 base pairs repeat, we have 2 or more adenine than thymine base pairs. Okay, for every 10, we will be having a 2 or more adenine. Okay, so this 2 or more adenine, wherever you look at this adenine thymine sequences, they are quite attached to the nucleosome cores. Okay, see here, and here. Okay, so here these areas, they are quite rich in the adenine and thymine sequences. Okay, and here also they bind with the nucleosome core. Okay, while the guanine and cytosine rich regions, they form the major and minor groups. Okay, here, and these guanine and cytosine regions, they are involved in binding of different types of transcription proteins, or gene activating proteins, or gene regulating proteins. Okay, so these kind of proteins, they mainly interact with this guanine and cytosine bases. Okay, which are present in the major group, and the adenine and thymine sequences, which are responsible for binding of the DNA with the nucleosome cores. Okay, so this is what facilitates the formation of nucleosome model inside the nucleus. Okay, and if we even read these points. Okay, so first point, runs of 2 or more 80 base pairs facilitate the DNA bending, while 2 or more GC base pairs have the opposite effects, that is the making the DNA in a linear way. Okay, so converting the bend form of DNA into linear form. Okay, when spaced at 10 base pair intervals. Okay, for every 10 base pairs, having 80 sequences like this. Okay, when spaced at about 10 base pair intervals. Okay, consecutive 80 base pairs help in bend DNA into circle. Okay, in this form. Okay, so they have to present this 80 clusters or 80 base pairs. Okay, so they have to be present in the nucleic acid chain for bending thing. Okay, for every 10 base pairs, they have to be present. Okay, and in the third point, when consecutive GC base pairs are spaced about 10 base pair apart. Okay, and offset by 5 base pairs from runs of 80 base pairs. Okay, DNA binding to the chromosome is facilitated. Okay, this is what about the nucleosome model. Okay, so GAC base pairs, they actually does the opposite things what 80 base pairs are doing in the formation of nucleosome model. Okay, so the GC base pairs are involved in attachment of lot of DNA binding proteins. Okay, and while 80 base pairs are involved in the formation of the nucleosome core particle. Okay, or the nucleosome model. So this is about the basic nucleosome structure. Okay, and if we talk about how the order will be like, how these histone proteins form the nucleosome model. Okay, first what happens is, the histone tetramer, see H3 and H4, okay. So they are deposited on the DNA molecule because they are rich in the basic amino acid constituents, okay. They get deposited on the DNA, and they form a site, okay. So this site marks for the assembly of this nucleosome core, okay. So this is called as the marking site, okay. So this is also called as the NCP, nucleosome positioning site, okay. So formation of this nucleosome position is very, very crucial for this nucleosome model, okay. And after formation of this NCP, then this H2A and H2B dimers, they interact, okay, and hold together, they form the octamer, okay. Then the linker histone binds to the nucleosome core, which is wrapped by DNA sequences, okay. This H1 seals the nucleosome positioning site, okay, or the nucleosome core, okay. Entry and exit site of the DNA, okay. So entry and exit site of the DNA, okay, in the octamer is sealed by what? Linker histone, okay. So the nucleosomes which contains the linker histones are often referred to as the chromatosomes, okay. And without the linker histones, that is just normal nucleosomes, okay. Remember this. So this is what the linker histones do. They ligate, or they are involved in the attachment of the entry and exit site, okay, like this. So this is what the major role of the histone proteins, okay. If the nucleosome contains the histones, you will be calling that as the chromatosomes, okay. And some more important points of the linker histones are given over here, okay. If you look at this, the fluorescence studies indicate that H1 molecules continuously dissociate and reassociate with chromatin. Together, the H1 protein and the histone octamer interact about 168 base pairs, okay. In some books it is given as 167 base pairs, okay. That is around 167 to 178 base pairs, okay. And H1 histone molecules can be selectively removed from the chromatin fibers by subjecting to preparation to solutions of low ion exchange, okay. What happens in the low ion exchange? The availability of divalent cations will be very low. Since the divalent cations are very low, the stabilization of the DNA will be very low, okay. Because of that, histone molecules can be removed from the nucleosome model, okay. So that is what happens in the case of low ion exchange solutions, okay. And then we also have the beads on zinc structures, okay. What happens if you remove this H1 histones completely from the nucleosome model, okay. So when H1 histones or the linker histone depleted chromatin is observed under the electron microscope, okay, without histones, okay, so it will be observed as beads on string, okay. So that means a string containing knots in between, okay. So these kind of knots is often referred to as the beads on strings, okay. These knots on the string, okay. The same kind of structure is observed under the microscope when you deplete the histone levels completely, okay. So this is called as the beads on string structure, okay, this one entirely, okay. And this beads on string also contains the histone proteins and also non-histone proteins, which we often refer to as the SMC group proteins or the topo isomerase 2 alpha proteins, okay. These are the non-histone proteins, okay. And we also have a concept called as the histone chaperones, okay. For depositing a typical histone onto the site of this NCP, we require a chaperoning or a courier proteins, okay, carrier or transporting proteins, okay. So these transporting proteins which transports or deposit the specific histone proteins on NCP site, they are often called as a histone chaperones, okay. Histone chaperones, what they do is they import or they transfer or they deposit the specific histone proteins onto the DNA sequences, okay. So this specific DNA sequences may form a NCP site, okay, or marking site, okay. So that is what the first point says about histones being highly basic in nature, are designed to bind variety of DNA sequences. However, it was discovered that octamer has got propensity to bind preferentially to certain DNA sequences, and this preference is mainly due to histone chaperones, okay. What these histone chaperones do? They take they first bind with the histone proteins, okay, and they transport or deposit these histone proteins to specific DNA sequences, okay. That is what the propensity, that is what the propensity that has got to the octamer core, okay. So this octamer has got a propensity to bind preferentially to certain DNA sequences, okay, on only certain DNA sequences, okay. The DNA sequences which are rich in adenine and thymine base pairs, okay. So at that sequences only the octamer is getting bounded. Okay, so this kind of preferential bounding is actually facilitated by what? Histone chaperones. Okay, not facilitating, but mainly due to this histone chaperones. Okay, for example, we have a nucleosome position in xenopause site for some 5s RNA encoding genes. Okay, these genes, okay, nucleosomes are also placed on on these genes. Okay, but the promoter site of these genes, they are left freely. Okay, so that means if you consider this as a nucleosome core, the core particle. Okay, so this is how the DNA gets wrapped over. Okay, understood. So this DNA might contain some kind of 5s RNA encoding genes. Okay, but the promoter of for these genes will be in this free nucleic acid sequence. Okay, the nucleic acid sequence which is left freely. Okay, the or which is a linker between the one nucleosome to another nucleosome. Okay, like this. Okay, so in this region the promoter for the 5s RNA genes are is present. Okay, so because of this preferential binding, okay, preferential or specificity. Okay, so specificity of this histone protein binding to certain sequences. Okay, so these histone chaperones are useful. Okay, like they play the major role in promoting this specificity. Okay, they play a major role in making the histone proteins to bind over specific sequences. Okay, as in the case of this xenopause oocyte. Okay, okay, so when the promoter is you know when the promoter is present in this free DNA or the DNA which is present between the two histones. Okay, so this promoter can readily bind with the transcription factor 3A and also the H1 binding. Okay, so and promote the H1 binding. Okay, so this is what the histone chaperones mainly do inside the nucleus. Okay, not only transporting, importing and depositing, they also add acetyl groups, and they are also involved in the gene regulating activities. Okay, so we have a lot of different histone chaperones over here. Okay, actually I have only discussed there are around 7 to 8 chaperones in this lecture. Okay, but we have lot more histone chaperones inside the nucleus, and some are under investigation process also. Okay, so first one is the AS1. Okay, so this AS1 actually act as a histone importer and histone transfer proteins. Okay, it transfers the histones to calf. Okay, first this calf takes the histone proteins from the ASF1. Okay, and then transfers to certain DNA sequences on the in the nucleic acid chains. Okay, in the nucleus. Okay, and increase the levels of this ASF1 is observed in the breast cancers. Okay, and I've also given a lot of histone chaperones and their roles. Okay, so these are the names for this histone chaperones. Okay, CF1 is actually called as a chromatin assembly factor 1. Okay, and along with the HIRA, what I gave you here. Okay, so this HIRA is also called a histone regulator A. Okay, so these are the specificity chaperones. Okay, preferential deposits H3.1 histones. Okay, this is H3.1 histone is a variant form of H3 histones. Okay, I've also I will be discussing these histone variants and their and their role in gene regulation in the next lecture. Okay, so they preferentially deposit the histone 3 variant, that is H3.3 and H3.3 and H3.1. Okay, so onto the DNA sequences. Okay, so this depositing is mainly done by this HIRA and CAF1 histone chaperones. Okay, and then we have the RTT106, which we also call as the regulation of tyrosine 1 transposition. Okay, the common name for this is okay, and this is involved in transferring an acetyl group onto the histone 3 lysine. Okay, so on the 56th amino acid, lysine amino acid. Okay, the 56th position the lysine amino acid gets acetylated by this RTT106 histone chaperone. Okay, I've said you right, not only depositing, importing and transporting things, histone chaperones are also involved in the acetylation and the deacetylation process. Okay, or the methylation processes. Okay, and then we have the AS1 which is also called as anti-silicin factor 1. Okay, so this is mainly involved in the transporting of a histone molecule or histone protein to CAF1 and a HIRA. And then we have the FACT is also involved in the gene regulation, facilitates active transcription, active chromatin transcriptions. Okay, so it is responsible for creating a DNA hypersensitive sites. Okay, and it also deposits H2A and H2B, which are histone 2 variants. Okay, H2 and H2B, normal histone, core histone proteins. Okay, so this fact is involved in the H2 and H2B deposits, and it is also involved in the gene regulation or transcription activities. Okay, So H2A, H2B histones, the core histones, they are deposited by this FACT, okay. And then we have the NAP1, nucleosome assembly protein 1, and which is a H2 and H2B chaperone, okay, again. And then we have a NASP, nuclear auto-antigenic sperm protein, okay. So this NASP is mainly involved in depositing or importing this H1 and H3 or H4 histones, okay, and also increase the turnover of this histone proteins, okay. And NPA1, which you also call it the nucleophosphine, and this is responsible for depositing the H1, H3 and H4, okay. The same roles played by this H1 and also this NASP proteins, okay, histone chaperones, okay. They quite have similar roles, okay. But this nucleophosphine, whatever the NPM1 is there, okay, so this nucleophosphine is actually involved in ribosome assembly as well as genomic stability, okay. So this is quite important point, okay. And then we have the ANP32E, so which removes the histone 2 variant, that is a H2AZ, okay, which is a variant form of histone 2. It removes the H2AZ from the chromatin, making the chromatin into active form, okay, or the making the chromatin get converted into euchromatin, okay. So this is what about the histone chaperones, okay. Then we also have the CH2Z1, which is a chaperone for H2AZ and H2AB, okay. So these are about the some different chaperones which are available in the nucleus, okay. There are a lot of other chaperones which are under investigation and which I don't know, okay. Then yeah, these are the major roles, okay. So and the nuclear processes which these histone chaperones are involved, okay. And the major chaperone or the histone chaperone is what FACT, okay, facilitator of active chromatin transcription, okay. So this is involved in the DNA repair mechanisms, replication, and also the transcription process, okay. So it actually ubiquitinates the lysine at 123, so 123 position, okay. So on histone 2b, so okay, we have a lysine at 123 position, okay. So it adds a ubiquitin molecule to this lysine, okay, and perform the post translation modifications on this histone 2b, okay, which is involved in the transcription regulation or the DNA repair thing, okay. And we have this we have the role of this FACT in this making the post translation modification on the H2X variant, okay, so which is also histone 2 variant, okay. So I'll discuss about the histone 2 variants and also their gene regulations, okay. For now, just remember this fact, these chaperones, they are also involved in the nuclear processes, that is transcription regulation or DNA repair mechanisms, okay, and also importing and depositing the different types of histones, okay. And then we have a next level structure, that is make further condensation of this nucleosome into a higher order called as a 30 nanometer fiber, which you often report as a solenoidal model or a simple chromatin model, okay. Now the next level chromatin compaction is beyond the 10 nanometer fiber, okay. So that is called as a 30 nanometer solenoidal model, okay. So 30 nanometer model or 30 nanometer fiber mainly gets organized into two forms, okay. One is referred to as the solenoidal model or also one start helix, okay, and the second one is referred to as the zigzag model or two start helix, okay. Both are 30 nanometer fibers only, but the arrangement of the nucleosome particles or nucleosome particles, okay, is different in the solenoid and the zigzag model, okay. So what happens in the case of solenoid model is the n+1 nucleosomes gets attached and form the solenoidal model, but in the case of zigzag model, n+2 so nucleosomes get attached and forms a zigzag model, okay. If we consider this, this is one nucleosome, okay, it will link with the second nucleosome, and this second nucleosome again links with the third nucleosome, okay. So it goes like this only, okay. So first to second, second to third, third to fourth, okay, and fourth to fifth, okay, and they form the solenoid model like this, okay. And when you look at this structure at top view, okay, so it will this structure will have a hole in it, okay. Understood? So this forms a solenoid or a cylindrical model, okay. You can easily call that as a cylindrical model, okay. But in the case of this zigzag model, the first nucleosome will get attached to the third nucleosome, okay, and the second nucleosome will now get linked with the fourth nucleosomes, okay. First with the 3 and second with the fourth, okay. So two n+2 nucleosome attachments will be seen in this zigzag model, okay. So because of this abnormal linking, the top view will be in this form, okay, fully covered form, okay. You won't be seeing any kind of a hole in between, or the cylindrical shape is not observed in this zigzag model, but the cylindrical shape is actually observed in this solenoid model, okay. So this solenoid model actually resembles the double helical nature of this DNA, okay. How the the 3D structure of the double helical DNA looks like, okay. The same way the solenoid model also be present in this nucleus, okay. So this is what about the solenoid and the zigzag model and their arrangement, okay, n+1 and n+2 arrangement, okay. And these are the two current models which are accepted by a lot of different molecular biologists, okay. So organization of this 30 nanometer fiber, okay, so it does not extend over entire chromosomes, okay, and it is punctuated by some sequence specific non-histone DNA binding proteins, okay. So apart from this histone binding proteins, okay, or histone proteins, we also have non-histone proteins, okay, or the DNA binding proteins, okay. So they bind to this 30 nanometer fiber. They further regulate the transcription activity, okay. So this 30 nanometer structure also seem to depend on transcription activity of the particular region of the DNA. Regions in which genes are being transcribed, okay, so they are apparently in a loss ordered state that contains little or if any histone 1 proteins, okay. So most of the gene inactive regions, they contain the histone 1, but gene active regions, they don't contain any kind of histone 1 proteins, okay. They are loosely attached, okay. And the 30 nanometer fiber, I've said you already, a second level of chromatin organizations, okay, and it approximately provides 100-fold compaction of the DNA, okay. Then we have non-histone proteins, okay. So the third level of proteins, which you also call them as second level of proteins, okay. The topoisomerase 2 is the second level protein, and the SMC group proteins is the third level proteins, okay. And if we even look at this, the topoisomerase 2 alpha, which is involved in the supercoiling of this DNA, which relieves the strain on the DNA by cutting the DNA strands, okay, by inducing the double stranded breaks, okay. And in mitosis it plays major role, okay, and it is concentrated at the centromere ends, okay, centromere and the axial regions of this chromosomes, okay. And if a mitotic chromosome lacks this topoisomerase 2 alpha, okay, the DNA will be in slim and elongated form, okay. That is exactly in the Z form of DNA, okay. The normal bDNA won't be present in this normal form, okay. So this bDNA, if the mitosis, if the mitotic cell, the cell which is involved in undergoing the mitosis process, if it lacks this topoisomerase 2 alpha, this bDNA will now look like Z form, okay. Okay, so this Z form of DNA is actually the thin and elongated form, or the slim and elongated form, okay. So this long and thin form of the DNA is mainly observed in the cells which lacks this DNA topoisomerase 2 alpha, okay. So mitotic chromosomes from cells lacking topoisomerase 2 alpha are long and thin, and the protein is thought to have rolling untangling DNA loops, okay. So yeah, I've said you right, the making by making this double stranded breaks, this topoisomerase 2 also untangles the DNA loops and condenses the chromosome axis, okay, and forms the chromosomes, okay, functional chromosomes, okay. And then this topoisomer 2 is also required for replicate a sister chromatid to separate one other during the mitotic anaphase, okay. So anaphase is what the splitting happens, okay, the chromosomal splitting happens, okay. At that phase, what this topoisomers do is it helps in separation process, okay. It aids in separation process, or it promotes the action of the separase enzyme, which will be seeing in this anaphase of this mitosis process, okay. And then we have the third category proteins, that is the SMC group proteins, okay. They contain 5 domains, okay. So out of these 5 domains, two are referred to as the Walker domains, okay, and one is a hinge domain, and two are alpha helical connector domains, okay. So these SMC proteins, they are non-histone proteins, okay. They actually contains 5 domains, okay. Two Walker domains, one hinge domain, and two alpha helical domains, okay. And these SMC proteins, they combine to form a heterodimer, okay. So these heterodimers may be different in different cells, okay, or in the different cell nucleuses, okay. So condensations coagins, they are also non-histone proteins, and they also contain the SMG proteins, but different types of SMG proteins, okay. One condenser may contain different types of SMG protein, and coagin may also contain different type of SMG proteins, okay. But they all referred to as the non-histone proteins, okay. And whenever these SMG proteins, whenever they combine, they form a V-shaped structure, okay, like this, okay. We have a head over here and also N-terminal over head over here, okay. So this N-terminal head and C-terminal head, okay. So they combine with the similar type of SMC protein to form a heterodimer, okay. So this is what referred to as the heterodimer, okay. So which contains two Walker domains, one hinge domain, and also the two alpha helical connector domains, okay. This is what the complete SMC protein will look like, okay. And then we have this bacterial SMC, which is made up of SMC protein and SMC protein, normal SMC protein, which you also call as a homodimer, because it contains same proteins, okay. Then we have the coercion proteins, which contains SMC1 and SMC, which is made up of SMC1 and SMC3, and also this Claisen family proteins, okay, which forms a complete ring-like structure during the condensation of the chromosomes, okay. All together, they form a ring structure and condenses the chromatin molecule further, okay, in the case of this mitosis, okay, at the end of the prophase, okay. So this coercion is made up of SMC1 and SMC3 along with the Claisen family connector proteins, okay. These orange proteins are often referred to as the Claisen family connector proteins, okay. Condensin protein also contains this Claisen family connector proteins, okay. And the condensin is actually made up of SMC2 and SMC4, okay. And then we have this DNA repair protein or DNA SMC group of protein which are involved in the DNA repair called as the SMCC5 and SMCC6 proteins, okay. They are made up of SMCC, SMC5 and SMC6 proteins, okay. And the coesins, okay, if you look at the roles of these coesins, okay, they are made up of SMC1 and SMC3, as I've said you. Coesins play a substantial role in linking together sister chromatids immediately after replications, okay, just after the replications of the interphase, S phase, okay. These coenzymes will come and bind with the sister chromatids, they link the sister chromatids, okay, keeping them together as the chromosomes condense into metaphase, okay. So during the start of the prophase, they combine with the sister chromatids and do the condensation process, okay. This linking is essential if chromosomes are to segregate properly to the cell division, okay. Obviously, so if the sister chromatins, if they don't get condensed, the lot of untangled thing will be happening in the case of this cell, okay, and the mitosis will be abnormal, okay. The coesins, along with the third protein called as a Claisen family proteins or connector proteins, okay, and are thought to form a ring, okay, complete ring around the replicated chromosomes, and that ties the sister chromatids together until the separations, okay. So this is what this ring is actually important, okay, and absence of this ring also involving a lot of abnormalities in the DNA, okay. Then we have the condensins proteins, which are also formed by this SMC group proteins only. SMC2 and 4 form the condensins, while SMC1 and 3 form the coesins, okay. It consists of SMC2 and SMC4 pairs, okay. The condensins are essential for condensation of the chromosomes, okay, as cells enter mitosis, okay. And in the laboratory, condensins bind to DNA in a manner that create positive supercoils, okay. And condensing binding causes the DNA to become overbonded, okay. So that means the supercoiling, okay. I've said you when I've discussed this supercoiling thing, okay, the major proteins which are responsible for the supercoiling or the overbonding of this DNA inside the eukaryotic genome are the condensin proteins, okay. And in the case of this bacteria, we have a lot of other different proteins called as the NAPs, okay, nucleotide associated proteins, okay. So this is what about, we also have this SMCC5 and SMCC6 proteins which are involved in the DNA repair, okay. So these are the SMC group proteins. In the bacterial group also, bacteria also contain the SMC group proteins, okay, which we often refer to as the non-histone proteins. Histone-like proteins in the case of this bacteria, okay. Majorly the archaeal family contains this kind of a SMC group proteins, bacterial SMC proteins, okay. And then the kinetochore, okay. So the kinetochore is the control center of this chromosome, okay, or the centrum in the centromere, okay. If the centromere or kinetochore is absent inside a cell, okay, the former two daughter chromosomes will not interact with each other, and they won't be participating in the mitosis process, okay. And this kinetochore is actually at the bilayer structure. Okay, bilayer in the sense it contains several layers. Okay, but it is distinguished into inner layer and the outer layer. Okay, so the inner kinetochore and the outer kinetochore. Okay, and if you look this kinetochore under the electron microscope. Okay, so this will appear as a several layers. Okay, and the inner kinetochore. Okay, the half part of the kinetochore is actually embedded in the centromere. Okay, actually it contained a special or specialized form of the chromatin molecule. Okay, the kinetochore and the outer kinetochore. Okay, so this contains the fibrous corona. Okay, so this contains outer plate with fibrous corona. Okay, and also the lot of protein complexes that link the chromatin molecule or the chromosome with microtubules. Okay, microtubules are the mitotic spindle proteins. Okay, or the spindle assembly checkpoint proteins. Okay, suppose if you consider this as the kinetochore. So this half part of the kinetochore actually contains centromere, and this centromere half part of the kinetochore is actually embedded into the centromere, and this centromere actually contains specialized form of the chromatin. Okay, like this. And this half part, okay, so it consists an outer plate with a fibrous corona. Okay, and also a lot of protein complexes which will be playing a major role in linking chromatin molecule with the microtubules or mitotic spindle. Okay, and major proteins which are found in this protein complexes or the outer kinetochore layer. Okay, so they are the centromere proteins. Okay, centromere A, centromere protein A, protein B, protein C. Okay, so these are the major proteins, and other 16 consecutive centromere associated network proteins. Okay, so they bind to this kinetochore, and they are present attached with this kinetochore throughout the cell cycle. Okay, so they don't leave the kinetochore or the this outer layer throughout the cell cycle. Okay, and the histone 3 variant, which we also call as the CNP3, CNPA is a histone 3 variant. Okay, so this is a majorly an inner kinetochore chromatin protein. Okay, and then we have the CNPB. Okay, which is a 17 base pair which binds specifically to 17 base pair sequence. Okay, and in alpha satellite regions. Okay, alpha satellites also they also present in the centromere regions only. Okay, so this CNPB specifically binds to the 17 base pair sequence in alpha satellites of the centromere DNA, and it is required for efficient kinetochore assembly. Okay, but a lot of roles are not known of the centromere protein B. Okay, and centromere protein B probably originated as the enzyme responsible for movement of ancient transposons. Okay, the T by 1 transposons. Okay, for the transposition movement, the CNB is responsible. Okay, and that transposon is actually present in the alpha satellite region only. Okay, and then we have the CNPC and CNPT, which are essential for DNA binding proteins. Okay, that bridge between inner chromatin and outer microtubule binding components. Okay, CNPC, CNPT, they actually link the KMN network. Okay, KMN network group proteins. Okay, which I'll be discussing in the next slide. Okay, so this diagram, yeah, this is actually an immunoblot. Okay, so this immunoblot actually got from a scleroderma patient. Okay, so when the antibodies of this uh, antibodies, if when the antibodies are raised in the scleroderma patient. Okay, so this immunoblot was developed. Okay, and this immunoblot actually represents the presence of this CNPA, CNPB, and CNPC proteins. Okay, CNPC links outer and inner kinetochore. Okay, and binds to CNPN and MIS2L complex. Okay, so this MIS2L complex is also a component of this KMN network. Okay, I'll tell you. And this CNPB binds to centromere DNA sequences and promotes efficient kinetochore assembly. Then we have the CNPA, centromeric histone, histone 3 variant. Okay, which is also a centromeric histone, and it marks site for kinetochore assembly. Okay, so it actually present inside inner side of the inner face of this kinetochore, and it marks the site of this kinetochore assembly. Okay, then comes the CNBB and CNPC and CNPT. Okay, so these are major centromere proteins. Okay, and remember the CNPA is mainly present inside inner layer of this kinetochore regions. Okay, and then we have the CNP C T. Okay, and yeah, one more point, it has been suggested that CNPC link is primarily involved in enabling chromosome movements, whereas CNPT linkages may monitor the status of kinetochore attachment. Okay, so this is what, one major point. So CNPC, it is involved in the chromosome movement. Okay, movement of this chromosomes. Okay, whenever the dynein proteins, they get attached or the microtubules attached. Okay, so the separation thing, that is regulated by this CNPC proteins, while the CNPT facilitates the kinetochore attachment with the microtubules. Okay, a set of proteins called the KMA network. Okay, which comprise the KNL1 protein, MIS12 proteins, and NDC protein complex. Okay, NDC80 protein complex further contains a lot of other sub proteins. Okay, so these are evolutionary conserved proteins. Okay, the core of mitotic kinetochore structure. Okay, and involved in the microtubule binding and the spindle assembly checkpoint function. Okay, so these are the KMA network proteins. Okay, so this entire group is often referred to as the KMA network. Okay, which contains NDC80 complex. Okay, and here we have the MIS12 complex, and then KNL1 protein, which is also responsible for this microtubule binding. Okay, and then we have here CNPT, CNPC, and also the HP1 protein over here. Okay, which is not shown in this diagram, and the CNPA, which is a histone 3 variant, is actually embedded inside the inner kinetochore layer. Okay, around here. Okay, CNPA, this is what the CNPA is. Okay, so these are the KMA network. Okay, so they contain a lot of other protein complexes. Okay, the best characterized outer kinetochore complex is what, NDC80 complex. Okay, which actually links with the microtubule. Okay, so this NDC80 complex, an elongated rod with globular ends linked by central coiled coils. Okay, and this NDC80 complex is a composed of NDC80P, NUF2P, SPC24P, and SPC25, because these are all subproteins which are mainly present in this NDC80 complex. Okay, so this is actually the NDC80 complex. Okay, and the KMA network. Okay, so the entire KMA networks, okay, in turn recruits other important microtubules and regulators. Okay, these regulators can be aurora B kinases, or the anaphase promoting complexes, or cell checkpoint kinases. So a lot of these regulators will be there. Okay, and microtubule motors, will be important microtubules motors. Okay, this will be dynein or kinesin proteins. Okay, so this kind of attachment is mainly recruited by this KMA network. Okay, so these microtubules and regulators, they include components of SAC apparatus. First is single spindle assembly checkpoint apparatus. Okay, which includes BUB protein, BUB-R1, MPS, MAD1, and MAD2 proteins. Okay, these are majorly involved in the mitosis process. Okay, and associated proteins such as RZZ complex. Okay, and proteins such as cytoplasmic dynein, which is a microtubule motor, and spindly, which is also kind of microtubule motor. Okay, class PS CNP. CNP is a kinetochore proteins. Okay, and the SKA complex. Okay, so they play major roles in establishing proper attachment and bioorientation of the chromosomes on the mitotic spindle. Okay, like this kind of orientation with the microtubules, is facilitated by this type of proteins. Okay, the microtubule motors as well as the regulators. Okay, once assembled during the prophase, the start of the prophase. Okay, the KMA network is present throughout the mitosis. Okay, it won't leave the kinetochore regions. Okay, whereas SAC, spindle assembly checkpoints, and the motor components are only abundant in the unattached kinetochores. Okay, that means the spindle assembly checkpoints, the protein which are involved in this checkpoint, they get diminished by the end of this anaphase. By the end of this anaphase, okay, you won't be seeing any kind of spindle assembly checkpoint, during the anaphase. Okay, and they are mostly abundant on unattached kinetochores. Okay, that during the start of this, during the start of this interphase, or the initial prophases. Okay, then we have this conserved MIS12 centromere complex. Okay, so this is actually linked to HP1 protein, heterochromatic protein 1. Okay, and outer kinetochore protein, ZWINT1 protein. Okay, I'll erase this. Okay, so this conserved MIS12 12 centromere complex. Okay, it is actually linked to heterochromatic protein 1 and outer chromatin protein 1, and it controls the assembly of outer kinetochore and it is ability to sustain tension. Okay, a lot of tension will be produced during this uh, separation processes. Okay, so this tension is actually controlled by this MIS12 centromere complex. Okay? And then we have this KNL1 subunit, which is also a component of this KMN network. Okay, and it is the major binding protein which recruits the BUB1 and BUBR1, which is a major proteins of this spindle assembly checkpoint. Okay, and by recruiting this, it also plays a major role in microtubule binding activity. Okay, so these are about the simple roles of this KMN network. Okay, which contains a lot of different proteins. Okay, this is actually for the depth level. And then we have the CNPC and CNPT independently link the inner chromatin and this outer NDC18 network. Okay, so this apart from the CNPA, CNPC and CNPT, they play a major role in linking with the microtubules. Okay, apart from this all other MIS12 centromere complex or KNL component, the CNPC and CNPT also links with the uh, microtubule molecules, microtubule proteins. Okay, sorry, they link the inner chromatin to the outer NDC18 associated network. Okay, or the NDC80 complex proteins. Okay, so this is what about the kinetochore proteins. And then we have this chromosomal DNA condensation, or the chromosomal organization in different phases. Okay, in the G1 phase, okay, in the start of this cell cycle, okay, the chromosome will be joining with this condensing proteins. Okay, and coercin proteins. Firstly, coercin proteins, they get attached to the sister chromatids, on top on below and top of the centromere regions. Okay, this coercin proteins, they after the replications. Okay, so in the S phase, we will be seeing this replication process happening. Okay, so after the replication, the sister chromatids will double their amounts. Okay, and becomes a two daughter sister chromosomes. Okay, so two daughter chromosomes. Okay, so these two daughter chromosomes, again got linked by this coercion molecules, which also contain this Claisen family proteins. Okay, the structure of the Claisen family is actually given here in this diagram. Okay, so this is how they are present. Okay, so these are actually the Claisen family proteins. This one, which has N terminals and C terminals. Okay, they are attached with the head domains SMC2 and SMC4. They form the coercion proteins. Okay, and these coercions, they condense further, condense the chromatids further. Okay, and then they recruit the condensing proteins for the further condensation. Okay, to make this compact, to make this large size protein, large sized chromosome into compact sized chromosomes. Okay, and the suppress enzyme, which is synthesized after the anaphase process, or sorry, during the anaphase process. Okay, after the separation of this the sister chromosomes, so called the daughter chromosomes. Okay, after the separation of the daughter chromosomes, and suppress enzyme is suppress protein is synthesized inside the nucleus. Okay, so this suppress protein will actually disconnects the Claisen ring with the coercion proteins. Okay, so that the total ring get disrupted, and it will it will exit out of the chromosome structure, leaving the two daughter chromatids separated. Okay, so now the two daughter chromosomes have got separated, and the condensing proteins, they also get exited out of the chromosome structures. Okay, so this entire separation thing and the condensation thing we actually detail be discussed in this cell cycle processes. Okay, cell cycle process and the regulations. Okay, but for now, just remember the roles of this condensing proteins and the coercion proteins. Okay, separase is what a protein which separates the two daughter chromosomes. Okay, and after removal of this condensing protein whole daughter, you will be having a two separate sister chromosomes. Okay, or sorry, not sister chromosomes, the daughter chromosomes. Then the actual condensation happens over here. Okay, so what we got is a 30 nanometer fiber. Okay, so after the 30 nanometer fiber, so in the interface, okay, we'll be getting only 30 nanometer fiber. Okay, and after the interface, the normal chromosomes, they normal 30 nanometer fibers, the cell cycle is promoted to mitosis. Okay, and in the mitosis, okay, if you observe this entire mitosis phase under the microscope, okay, and this mammalian chromosomes, they condense in the prophase only. Okay, the prophase is what the condensation phase. Okay, and in the initial period of prophase, the 30 nanometer fiber folds into a 100 to 130 nanometer chromonema fiber. Okay, first condensation is what, folding a 30 nanometer fiber into chromonema fiber, and then the disappearance of a nuclear envelope also happens. Okay, and then this chromonema fiber fold into structure with a diameter of 200 to 50 nanometers, which we call that as a middle prophase chromatids. Okay, and by the end of the prophase or the initial metaphase, okay, so which this middle prophase chromatid further folds into 500 to 750 nanometer in diameter, forms the chromatids. Okay, these chromatids are actually observed during the metaphase, that is just after the end of this prophase. Okay, and at this metaphase, when the nuclear envelope retracts into endoplasmic reticulum. Okay, so that means whatever the nuclear envelope has got synthesized from the endoplasmic reticulum initially, so that will now get disappeared, and this disappearance is actually due to the retraction. Okay, so that means the entire nuclear envelope goes back into the endoplasmic reticulum, okay, and converts into vesicle forms. Okay, so this vesicle now contains the nuclear envelope. Okay, and then ultimately the full length of the two associated daughter chromosomes generated by the DNA replications during the S phase. Okay, S phase is what, the synthesis phase, the DNA replication phase. Okay, so there the two daughter chromosomes have got generated. Okay, these two daughter chromosomes, now they get condensed and get linked by a central construction called as a centromere. Okay, so this centromere links the two daughter chromosomes, okay, and makes the complete double sized chromosome structure. Okay, so this chromosome structure, if it enters into the anaphase, it gets separated into again two daughter chromosomes. Okay, and cell division happens. The two daughter cells will be formed in the end of mitosis cycle. Okay, so this is what the overview of this chromosome package again. Okay, the first level of the DNA, the nucleosome structures. Okay, the nucleosome structure will further condense and they form the loop, chromatin loop, with the help of the scaffold proteins. Okay, and this entire chromonema or the 100 nanometer fiber will further condense to form a 250 to 300 nanometer fiber, which we also call as a chromatid middle phase chromatid fiber, and then it further condenses to form the chromosome. Okay, so this is what about the actual genome organization in the eukaryotes. Okay, but the prokaryotic DNA doesn't undergo this kind of organizations. Okay, so since they have a little amount of the DNA when you compare with the eukaryotic genome. Okay, so they only involve different types of proteins. Okay, like nucleus associated proteins and some kind of a non-histone proteins such as SMC group proteins, SMC and SMC homodimers. Okay, and by using these kind of proteins, they organize their own genome, okay, into a compact sized chromosome, chromatin like material, or the nucleoid material. Okay, and this will be fitting into this bacterial cells. Okay, and certain archaeal family, they contain this nucleosome like structures in them. Okay, which is involved in the compaction of the DNA molecules. Okay, and the presence of this uncompacted DNA inside the bacteria, okay, is mainly responsible for the very easy conjugation processes, and regulating the transcription and the easy replication processes. Okay, so this is what about the prokaryotic DNA. I gave you the important points over here. Okay, and this is about the today's class, the genome organized and the packing of this DNA into compact sized chromosomes. Okay.